You know, we usually think of our DNA as this fixed, stable blueprint for life. But what if I told you it's more like a living document, one that's constantly being edited? That's what we're diving into today, the restless, dynamic world of the genome. And the stars of our show? These little things in bacteria called mobile elements, or, as you might know them, jumping genes.
So just think about that for a second. Is your genetic code really this unchanging manual set in stone the moment you're born? For the longest time, that's exactly what science thought, this neat, orderly library of instructions. But it turns out the reality is way more chaotic and honestly a whole lot more interesting.
Yeah, because it turns out there are these secret editors hiding in the code, transposable elements. You can picture them as these little snippets of DNA that can literally cut themselves out of one spot and paste themselves into another, rewriting parts of the genetic story as they go.
Okay, so to really get a handle on how this all works, let's meet the main characters in this genetic drama, and we're going to focus on what's happening inside bacteria.
So these jumping genes really come in two main flavors. On one side, you've got the simple insertion sequences, or IS elements. Think of these as a basic, no-frills moving kit. They only pack the absolute essentials they need to move themselves. But then you have the more complex transposons, or TN elements. These guys are like a moving kit that's also hauling a ton of extra luggage, and that luggage can be some pretty serious stuff, like genes for antibiotic resistance.
Let's zoom in on those insertion sequences for a moment. They are the true minimalists here. Their design is just brilliantly simple. You've got a single gene that codes for an enzyme called transposase, and it's sandwiched between two short DNA sequences that are perfect mirror images of each other. We call those inverted repeats, and they basically act like little handles for the enzyme to grab onto when it's time to make a jump.
And that brings us to the real MVP of this whole operation, transposase. This enzyme is the engine that drives everything. It's like a molecular pair of scissors and a tube of glue all in one. It recognizes the element, cuts it out, and then pastes it into a brand new spot in the genome. Seriously, without transposase, nothing is going anywhere. Now, even the more complex transposons have different ways of getting things done. You've got composite transposons, like the famous TN10. These are basically a piece of genetic cargo, let's say an antibiotic resistance gene, that has hijacked two IS elements on either side to act as its moving trucks. Then you've got non-composite transposons, like TN3. These are more self-sufficient. They've got the cargo and the engine all built into one sleek package. All right, so we've met the players. Now for the action. How do these things actually jump? What are the mechanics of this little genetic dance?
But before we get into the two main ways they move, there's a key piece of evidence that's always left at the scene of the crime, so to speak. Whenever a transposon inserts itself somewhere new, it actually creates a tiny little duplication of the DNA sequence right at that target spot. We call these target site duplications, and they're like molecular footprints, a dead giveaway that a jumping gene was there.
Okay, so the first method is called conservative transposition, but it's way easier to just call it cut and paste. It's pretty straightforward. First, the transposase enzyme gathers at the ends of the element, forming this little protein machine called a transposome. Then that machine literally snips the entire element clean out of its original spot on the chromosome. The whole thing then moves to a new location and gets pasted in. The cell's repair crew comes in to patch up the old spot, and that's it. One element, one move. Simple.
Now, the second method is much sneakier. We call it copy and paste, or replicative transposition. Instead of cutting the element completely out, the transposase just nicks the DNA and links it to a new target. This clever move actually tricks the cell's own DNA copying machinery into making a brand new copy of the entire transposon. For a brief moment, this creates this giant fused DNA molecule called a cointegrate. Then a second enzyme called resolvase comes in to, well, resolve the situation. It snips the cointegrate apart, and voila, you're left with two separate DNA molecules, and each one has a copy of the jumping gene. The element gets to move and multiply.
So just to be super clear on this cointegrate thing, it's this really fascinating temporary structure where the original DNA and the target DNA are literally fused together with a copy of the transposon at each of the two junctions. It's the absolute hallmark of that copy and paste method, and it's how the element manages to increase its numbers.
So, we've seen who the players are, and we've seen their moves. But what's the point? What does all this jumping around actually do? Well, this is where we get to the real-world impact, and it's a story of both incredible creation and total chaos.
So why should you care about any of this?
Because this isn't just happening in some obscure bacteria. It is happening inside of you, right now. Get this, roughly half, and let that number sink in for a second, 50% of the entire human genome is made of these jumping genes, or their ancient fossilized ancestors. They are a massive part of what makes us who we are.
And their impact is a true double-edged sword. On one side, if a jumping gene lands right in the middle of an important gene, it can cause a harmful mutation and completely disrupt how a cell works. But on the flip side, all this constant shuffling of the genetic deck is an incredibly powerful engine for evolution. It creates new genetic variations, the very raw material that drives innovation and allows life to adapt. And maybe the most urgent and terrifying consequence of this process today is its role in spreading antibiotic resistance. The rise of the superbug is, in so many ways, a story that's being written by jumping genes.
Here's how it goes down. A transposon with a resistance gene can just hop from the main bacterial chromosome onto a plasmid, which is a small, circular piece of DNA that acts like a portable flash drive. That plasmid can then be passed from one bacterium to another in a process called conjugation. And just like that, the resistance gene spreads like wildfire through a population, even jumping between completely different species of bacteria.
Okay. All of this raises a pretty obvious question, right? If this whole process can be so dangerous and disruptive, how do organisms stop it from spiraling completely out of control? There has to be some kind of regulation, a way to maintain this fine balance.
I mean, it's a fair question. With all these elements jumping around, why isn't the genome just a chaotic, mutated mess all the time? Well, the answer is that life has evolved some incredibly clever ways to keep these jumpers on a very tight leash.
The main strategy is to control that transposase enzyme, the engine of the whole process. And there are a few ways to do it. Some elements, like TN3, actually make a protein that physically sits on the DNA and blocks the transposase gene from being read. Others, like TN10, use this amazing trick with something called antisense RNA. They make a tiny piece of RNA that intercepts the instructions for making transposase, stopping it before it can even become a protein. And some, like TN5, will even produce a faulty, decoy version of the enzyme just to gum up the works of the real one.
And what's so elegant about that antisense RNA system is that it creates this perfect little feedback loop. The more copies of the TN10 element you have in a cell, the more of this antisense RNA gets made, which in turn slams the brakes on any more jumping. It's a beautiful, self-regulating system that keeps the element from completely taking over its host.
Which brings us to this final, really fascinating question. What are these things? Are they just selfish genetic parasites only out for themselves? Or are they the essential, chaotic engines of evolution, providing the variation that life needs to survive? The truth, like it so often is in biology, is that they're both. They are the source of chaos, and they are the spark of creation, all at the same time.